Today we are talking about the DNA structure and how each part interacts with each other. First, guanine always pairs with cytosine with 3 hydrogen bonds, and adenine always pairs with thymine with 2 hydrogen bonds in a process called base pairing. This is because these structures are most stable with each other with respect to hydrogen bonding. Hydrogen bonding is not real bonding, but is attraction between hydrogen, oxygen, nitrogen, and fluorine. The pyrimidine bases, both adenine and guanine, have a single ring in them. The purine bases, cytosine and thymine, are heterocyclic, which means they have 2 rings in them. Base stacking is how the base pairs are ordered, with one on top of the other, roughly 340 picometers apart. The phosphate backbone is made of alternating deoxyribose, which is a sugar, and phosphate groups. The sugars are what hold the base pairs in place, and the individual carbons are very important in interactions. For example, the 1' carbon is where the nucleobase attaches. The 2' carbon is there to maintain the shape and structure of the sugar and the DNA. The 3' and 5' carbon are there to link to 2 different phosphate groups. These phosphate groups will attach to more deoxyribose, and the cycle repeats. The major and the minor groups are both the same thing, but one is a larger gap than the other. The major groove is because of the negative charges on the phosphate group. This is taken advantage of by proteins specific to either the minor or major groove. However, there are many more proteins that interact with the major groove because there simply is more space for them to interact with. DNA is made of anti-parallel strands, which are parallel but lead in opposite directions, because proteins that aid in creating complementary strands create leading ends, the one that's further along, and lagging ends in replication forks. One side is 3' to 5', while the other is the opposite. Handedness is in which direction DNA turns, and the vast majority of DNA is right-handed. This is also caused by negative charges of the phosphate groups. The ions in the salt in saline sodium citrate reduce the negative charges in DNA. This would cause the DNA to bind more tightly, becoming more stable. Like with greater guanine-cytosine pairs, it will likely increase the melting point of the DNA. In addition, it will impair the functions of the major groove proteins because the major grooves will shrink while enhancing the minor groove proteins. It could also cause less tension to be put on the DNA, causing supercoils to be much larger or even non-existent. This is my citation.